Hello, my name is Thomas Baumgartner, and this is
Jason McCormick.
and we're the co-managers of the flow cytometry core facility. Today we're going to give you a brief talk about what flow cytometry is and introduce you to the services we offer at our facility.
Okay, well, this is us. We're the flow cytometry core facility. If you already know something about flow cytometry, you'll probably understand why we have this slide. If you don't, what is flow cytometry? Well, flow cytometry is analyzing cells. It's very fast. Generally, it uses fluorescence. There are imaging and mass spec cytometers around nowadays. They're quite cool. We're looking to get some of those in the core. Historically, there's impedance. That usually is involved with counting and measuring volumes of cells. But we like fluorescence, and most flow cytometry data is fluorescence-based. Main reason is that you have resolution from a few to a few 100,000 molecules of label per cell. It's high parameter, or what they're calling now high density, so you get multiple parameters per cell per time. You can also physically separate the cells. That's called sorting. And a little note, you may have heard the term fax. This is not a fax machine, if anybody still uses those. It's really not fax either. It isn't fluorescence activated cell sorting. There's a lot more to it than that. Historically, flow cytometry as we know it got started by the US Army. There was a big interest in knowing whether or not our enemies were trying to kill us with biological weapons on the battlefield, and the Army came up with a device that looked and acted like what we have in the labs today. Shortly thereafter, the impedance flow cytometer was patented. That is still used today in hematology analyzers and in a device called the Coulter counter, if you're familiar with one of those. We got the first sorter in around 1967. In the 70s, Caesar Milstein came up with monoclonal antibodies and the ability to make hybridomas. Around the same time, BD Biosciences, then called BD, came out with the first sorter, which is called FAX1. That's where we got FAX. In the 80s, HIV research and the commercialization of benchtop flow cytometers wound up with flow cytometry becoming the assay for doing HIV research. Since then, we're now up to something like commonly finding 5 lasers and up to 20 or more fluorescent colors in a flow cytometer. So how do you do flow cytometry? I want to say just read the slide, but basically, read the slide. You have to know a lot about a lot of things. You have to understand the cells, you have to understand the reagents, you have to understand the electronics, and you have to know how this all fits together in order to get some meaningful data out of this. I also recommend Howard's book. It's out of print, but you can download a free copy. So why do we do flow cytometry? Or how much do we do flow cytometry? If you look at this chart, a simple PubMed search on those terms at the bottom, flow cytometry or FAX, is in the red. Last year, there were over 10,000 entries citing flow cytometry. Contrast, we have sequencing in the blue curve, and I've drawn a little dotted line there for next-gen sequencing. Once that hit, things went kind of crazy. So where do all those publications come from? Well, all of the ologies. Basically, if it's got cells and you can tag it somehow with a fluorescent tag, or maybe even take images of it, you're going to do flow cytometry at some point. Some of the assays that are common, phenotyping, so are they CD4, are they CD3, is it a B cell, is it an NK cell? You can measure soluble analytes, that's a bead-based ELISA. You can look at the amount of DNA in a cell. That was sort of the original application of flow cytometry. If you want to count stem cells, you want to look at platelet status. If you want to do drug efficacy, if the assay is fluorescent or if it can be done in cellular suspension, a flow cytometer can do it. And if you can do that, you can get the ones you want back at high purity with good recovery using a flow sorter.
And now let's cover what we do for you in the core. What can we bring to your research? Our mission statement, we are your partners in flow cytometry. We're here to provide you with the best equipment, education, and service. Our services basically break down into two categories. We have an extensive training program. So if you don't know anything about flow cytometry, we're here to teach you, consult with you, and really help you get your experiments off the ground. So we'll take you from being a novice to being an expert. We have kind of two arms to our services. We have the sorting service, which is operator assisted. You're never actually touching the instrument yourself. For that, you're not required to take a training course. You just simply have to consult with us. We'll help you set up your experiments, and then we're going to get you started sorting. If you're going to be doing analysis only, we offer a variety of analytic instruments, and on that arm, we also offer training. Our course is a 4-hour course with a 2-hour hands-on session on our newest and most advanced analyzers, and they'll get you up and started. I would also like to mention that even if your lab has its own flow cytometers or using flow cytometers somewhere else, we are here to be a resource to the entire community. So please feel free to come talk to us, take our courses, take advantage of our expertise, even if you use equipment that's not in our facility. And briefly, let's summarize the equipment we have available. Some of you might be familiar with these. We have 2 sorters in our facility, a BD Aria with 5 lasers and a BD Influx with 6 lasers. They also have a bunch of parameters, so we're pretty much able to do anything you can think of in flow cytometry on these 2 instruments. And we have a variety of analytic instruments. We've been adding analytic instruments steadily the last couple of years as the need arises. Our most advanced instrument at this time is a BD LSR4 Tessa, again with 5 lasers and 18 parameters. And thank you very much for your attention today.